Well,
Morning.
Hi.
morning. Hi. So, Paul, how are you? How are things on your end?
Things are fine. I have no major updates.
Okay. Yeah. So I guess we can get started. So I have a couple of papers here on sort of self-reproduction and minimal chemistries. I think one of them was pointed to by Hussain. I think it was in the Slack where he pointed out this paper. It's a new paper on the origin of life. There's also the paper on the minimal chemotactic cell, and then a paper on reaction networks in thermodynamic space. So I'm going to go over those and talk about their implications and things like that. Okay, so the first paper is this new paper. It's called Self-reproduction as an autonomous process of growth and reorganization in fully abiotic, artificial, and synthetic cells. So this has a lot of relevance to early life stuff, and especially some of the things we've been talking about in this group off and on for the last couple years. So their interest is in sort of thinking about self-reproduction and how to build a model of that, or a minimal model of that. So when I say minimal model, you try to build a model with the minimal components that makes a system work. You don't try to overcomplicate it, but it should demonstrate, the mechanism of what you're trying to achieve. So for an early life model, we have these minimal models of cells, which are these vesicles, which are these just basically empty containers, and then we put things in the container to try to initiate biochemical reactions and some of these behaviors that you see in very simple cells. And so, we I guess associate with life things like reproduction, being able to carry a genetic code. And of course, there were RNAs out in the world before there were cells. But one of the things about the cell, which is modeled by this vesicle, is that you can do that within a container and it protects it from the environment and it has its own sort of self-containment. So this is the kind of the background as to why this is a really interesting problem. And then, of course, the self-organization of this process, meaning that it comes from basically from no real template. There's no earlier form of this. So, when cells evolved, there was no cell to come from. It just had to kind of self-assemble on its own. And this happens, of course, again with eukaryotic cells, where you get organelles in the cells, inside the cells, and you make this jump in this transition in evolution. So the significance of this paper is self-reproduction is one of the most fundamental features of natural life. So this is again stating this like basic fundamental thing about cells and natural life and so on. This study introduces a biochemistry-free method for creating self-reproducing polymeric vesicles. So they're actually interested in this biochemistry-free method, which they'll describe in here. In this process, non-amphiphiolic molecules are mixed and illuminated with green light, initiating polymerization into amphiphiles that self-assemble into vesicles. These vesicles evolve through feedback between polymerization, degradation, and chemoosmotic gradients, resulting in self-reproduction. So you have this vesicle, you are putting in these non-amphiphiolic molecules, you're activating them with a green light or you're illuminating them, and then this initiates polymerization, and then this creates these amphiphiles that self-assemble, and then this self-assembles into vesicles. So we start with no vesicle, we start with these non-amphiphiolic molecules, and we build these steps to these vesicles that we're interested in. And so they identify the steps needed for that. As vesicles grow, they polymerize their contents, leading to their partial release and their reproduction into new vesicles, exhibiting a loose form of heritable variation. So again, this is not like the kind of cell that we see today. This is a minimal model of a cell, basically doing this thing. And, I guess we can make parallels between the cells of today and this model, but the idea is just to sort of give a rough approximation of the process. This process mimics key aspects of living systems, offering a path for developing a broad class of abiotic, lifelike systems. So, of course, we've talked about, in the past, we've talked about minimal cells, we've talked about some of the programmable materials and things like that. So this kind of fits into that area. I mean, there are reasons why you would want to do this other than sort of looking at the origins of life. You'd want to do this maybe for some bioengineering application, or things like programmable matter, etc. So this is the abstract. Among the characteristics shared by all extant living systems, their chemically controlled self-reproduction ranks perhaps as one of the most spectacular and exclusive. Under the control of their internal chemical networks and in tight relationship with their environment, from where they harvest food, energy, and information, natural living systems spawn representations of themselves, or reproductions. That's how they define reproductions, as these representations of themselves, as fully functional systems, which also autonomously and under sufficiently similar conditions to those of their progenitors, are again capable of self-reproduction in new generations. Indeed, when living systems reproduce, their species continues to exist via their progeny and propagates as such into the future, also enabling fundamental aspects of their Darwinian evolution due to the generation of reproduction of populations, which include heritable variation. So we need to make that step up to something that can undergo Darwinian evolution, have heritable variation that's heritable across cells, and be able to create new generations that can be selected and things like that. So that's kind of, but we have to start with building a structure for that, a container for that. And again, I want to stress that these aren't like actual cells that you see today, because the actual cells we see today, the membranes have a lot of things going on. There are pores and receptors on the membranes, and the membranes are made of, complex materials with multiple different molecules contributing to the surface. So we don't really have any of that. We just have a base container, and then we're loading things into it, or there are things that are going into it. So it's like a bag of reactions, basically. And so in extant biochemistry-based life, even for the simpler single-celled living systems such as bacteria, the process of reproduction is complex and involves many complicated and coordinated steps. So this is, again, when we see reproduction in cells today, there are a lot of steps to that, like to a cell division, or things like that. So we're not really modeling much of that here. These take place within the cell under the precise control of biochemistry and collectively constitute the cell division cycle, or CDC. So, of course, the cell division cycle is initiated by the expression of a number of genes in sequence, and of course, you have different phases under which the cell cycle operates, and sometimes those phases are expanded or shrunk down, and they can affect the division dynamics of the cells. So you have this basically this genetic regulatory network in modern cells that controls cell division cycle. So cells can proliferate, or they can not proliferate in a given context based on that network. Now, we don't have that in vesicles, but we just want to see some sort of reproduction. And you can imagine in early life, you had some mechanism for reproduction that didn't involve the cell cycle. And then eventually the cell cycle emerged in the cell. So we're not really answering that question directly, but we're recognizing that the biochemistry, when they say biochemistry-free, the biochemistry they're referring to is something like the cell cycle, where there's a initiation of a gene expression, and that constitutes a gene regulatory network, and those things are regulated in time, and they produce this coherent output of cell division. Like Virchow said in 1858, omnis cellula e cellula, or every cell comes from a pre-existent cell. So this is, again, with respect to, say, like bacteria or eukaryotic cells, that cell division comes from previous cells, and then those cells are the product of like, developmental precursors to that. But we're always kind of coming from a pre-existing cell, a pre-existing template. Okay. However, we may ask, is extant biochemistry, with its delicate and well-tuned complexity over billions of years of evolution on Earth, necessary for a protocellular chemical system to reproduce? So the protocellular chemical system is the thing they're going to focus on here. When they say, they're talking about the delicate and well-tuned complexity of biochemistry, they're talking about these processes like cell division, where we have the biochemistry in place. We don't need to reinvent the wheel to see how that works. But in these vesicles, we don't have that. Can one construct in the laboratory non-biochemical compartmentalized chemical systems capable of autonomous self-assembly and self-reproduction? And of course, in this case, they're going to try to answer that question with perhaps, and then build this model. Answering these basic questions is important for systems chemistry, artificial life, or for protocell research seeking to understand what life may have been like before the advent of biochemistry, or last universal common ancestor, or LUCA, during a so-called protocell era, and for understanding the potential for life in the universe, including exoplanets. So this is just to say that this is relevant to a wide number of topics in like systems chemistry and in the origin of life. During the protocell era, life must have been simpler. And they have citations for that. This is, I guess, a quote from one of these papers. And hundreds of millions of years could have elapsed, evolving the transition from chemistry to some form of generalized life. So basically, before you have, I guess, generalized life, which I assume is something to do with cells, you have this chemistry that sort of, free-floating chemistry, where you get these reactions and you get these things that are maybe on the edge of life, and then this kind of becomes what we might call generalized life at some point, and that leads us to our last universal common ancestor, which is where phylogeny really began, where you started to get cells that divided, and then you had changes to those cell populations, and then you started to get different taxonomic differences between them, and you'd have it was basically the start of the tree of life. So that's kind of, and then that area is very murky in terms of what the steps there look like, but that's what they're trying to get at here. Finally, these questions are also central in the old but nascent field of synthetic artificial life, based on non-biochemical material implementations of fully synthetic chemical systems capable of mimicking natural living systems. And there's some citations here. So, there's this area, like we know the Artificial Life Conference, and they focus a lot on computer simulations. But there's also this area of artificial life where people go into the lab and try to create these conditions for, some sort of origin of life scenario. So people use vesicles. Of course, they're the Miller-Urey experiments that involved taking a bunch of chemicals and putting them in a soup, and then animating them under certain conditions with electricity, and then getting different chemical reactions to happen. And so there's a history of that whole paper, the Miller-Urey paper. The original paper And then there were some replications that were done later with some modifications. And then there was a sort of a revisitation of that paper by the grad students of Miller and Urey, and they were able to get a much more robust result with modern equipment. This happened about 10 years ago. So, there's this literature that they're building on here. In an effort to explore the answer to the above broad questions about the necessity of biochemistry, perhaps helping to trace back life's origins to the combination of carbon chemistry and the physics of self-organization, and building upon the opportunities in precisely this direction offered by polymerization-induced self-assembly, or PISA, and that's something that is in the literature. And so in this case, they have designed a PISA batch reactor where an aqueous solution, and strictly avoiding any biochemical molecules, we synthesize amphiphiles that self-organize, self-assemble, and self-boot into chemically active micelles. Okay, so yeah, they use boot in the way that we would use the term boot to talk about loading a program into a computer. So that's what they're, this is of course a very strong parallel with a lot of the engineering applications that maybe they seek to do, use this for. The micelles then develop into functional giant vesicular systems, or GVs, from just a few small and low complexity molecular species chosen ad hoc for us by implementing PISA. And so they start with a homogeneous blend of several simple molecules, and then they basically exclusively rely on chemical forces controlling the exchange of atoms among molecules, and then use light as a source of external energy. So that was that green fluorescent light that they were using. They're using that as a source, so they basically make it a closed system. So the only source of energy they're getting is the light. The aqueous thermostated and illuminated reaction mixture contains a hydrophilic polymer with a small and slightly hydrophobic chain transfer agent molecule, or CTA, bonded at its end, plus some selected monomers such as acrylonitrile or hydroxypropyl methacrylate, or HPMA. And this can be photopolymerized to the above hydrophilic polymer to yield amphiphylic block copolymer molecules. And finally, the PISA mixture also contains at least one photocatalyst, or inverter, a photocatalyst molecule that acts as an initiator, a chain transfer agent, and a terminator of the polymerization reaction. So they're doing this sort of polymerization, the steps of polymerization in this chemical system. And so note that this very simplified out of equilibrium system can abstractly be imagined as a laboratory version of a warm little pond where the notion of heterotrophic abiogenesis can be explored. So this is, again, this idea of that, life originated in warm ponds where you had, energy fluxes coming in, like lightning, or, light energy, like UV energy, back when the Earth didn't have the UV protection that we have now. And so that was sort of the driver for the origin of life. Okay, so they go through, they're talking about how they switch on the illumination. They're able to get these results, and they have, concentration. So as the concentration of the amphiphile increases beyond its critical micelle concentration, the activity of the chemical reactions leads to liquid phase separation due to the ongoing polymerization process of the hydrophobic block in the amphiphiles, which eventually self-assemble and self-organize into micelles. So they go through these steps of self-assembly, and so during their self-assembly, in the course of the living polymerization synthesis of the amphiphiles, these micelles entrap some of the current reaction medium, which guarantees the continuation of the polymerization reaction in their interior, although at a rate different from the one in the bulk, which consequently generates and amplifies osmotic gradients. So we're basically, by simultaneously changing the packing parameter value, of the living, and in this sense of living polymerization, amphiphile due to the increase in the length of its hydrophobic block. The ongoing internal photopolymerization reaction also powers the dynamical morphological evolution of the micelles, which develop in time and become these micron scale vesicles. So there's very tiny vesicles, and they're self-organizing in this way. Okay, so that's a little bit about the background of the paper. And this is a figure, this is figure 1. So this shows, maybe puts a little more familiar face on what's going on here. So we have A here. This is an illustration showing the different stages of polymer vesicle growth, leading to the action of expulsion of amphiphiles. So this is where we have, this is the original, the starting point here. We get polymerization and growth in size. So we get this vesicle that grows in size, and then these amphiphiles are ejected from this, and that's what this is here. So we have an illustration showing the different stages of polymer vesicle growth here, leading to the action of expulsion of amphiphiles. Okay. And this is a key to the different molecules that we have here. And the green light, of course, is in green, so you stimulate this with green light, it grows, and then you get these amphiphiles. And then B is the illustration showing the formation of new vesicles from the reorganization through self-reproduction of amphiphiles expelled into the bulk. So in A, we see this process of stimulating the original vesicle with green light. We get the growth, we get the expulsion of amphiphiles into what they call the bulk. And then, of course, there are a bunch of these in sort of in this medium. So you have, in this case, 7 of them or 6 of them, and they're all expelling amphiphiles into the bulk, and those amphiphiles aggregate, and then when they get to a certain density, they form a new vesicle, and this process can start over again. So they're actually self-reproducing in this way. So this is the synthesis of amphiphiles in polymer vesicles using raft polymerization. That's here, where you have this agent, you have raft polymerization, you have the amphiphiles, they self-assemble into these piece of mixtures. B is a filtration of piece of mixture and subsequent irradiation of amphiphiles and vesicles separately. So you see this where you have the piece of mixture, you have this filter, you have the filtrate, which are the amphiphiles, you have the retentrate, which are the vesicles. You stimulate them under a microscope and you count the vesicle number growth for the vesicles. For amphiphiles, which are filtered, you stimulate them with green light, and there should be no vesicles. So you're able to basically purify the vesicles in the sample. And then C is a plot showing the growth in the number of vesicles during irradiation of retentrate with green light. So this is showing the number of vesicles that increases over time, but then plateaus at about 5,500 vesicles, and just showing that you can get certain critical number of vesicles in the mixture. And then D is the optical image of the filtrate sampled irradiated with green light, showing no visible objects in the image frame. So the filtrate is this, or the amphiphiles, and they shouldn't have vesicles in them, and indeed they don't have anything in them, so it's clear. So you see what's going on here. We have this process of reproduction. We have this process of self-assembly from amphiphiles that have been expelled from previous vesicles. And then we have this piece of mixture, which we put through a filtrate. We separate out the amphiphiles from the vesicles. We count the number of vesicles, and then we make sure that there are no vesicles in the filtrate, showing that those things can be separated out. Okay, so that's this. And so this paper, I think Hussain put this in the Slack, where there was a lot of excitement around it as being like this key paper in early life research.
Yeah,
and
pretty bold to call it the most important paper of 2025. That's a bold one, eh? I mean, our model that we are studying with me, Dr. Gordon, and Savino, we are doing excuse me, our work is our work is empirically based, so that's not the same one as what we were talking about, but yeah.
Yeah, there are a lot of different ways you can get at this problem. A lot of ways you can get at it is with simulation, basically simulating a hypothesis, because it's really hard to get at what actually happened because we kind of have estimates of what the conditions were like, but all we can do are try things and see what works in the lab, and then see maybe what works in simulation and try to put those together. So. Okay, so that's, I think, enough of that paper. The second paper I wanted to talk about is this paper. This is from Science Advances. This is what they call the minimal chemotactic cell. So this kind of leads from the last paper, but it talks about things that you find in minimal cells that exhibit what they call chemotaxis. So chemotaxis actually is quite important to C. elegans, and it's the main way in which a lot of organisms and single cells will try to find their way in the world. So what taxis are responses that an organism will give to some environmental stimulus. So a good example of this is a phototaxis, where you have like a source of light and it's at a certain location. And that light, of course, there will be like a gradient of light from the source that decreases with distance. And then you'll have like a cell or a small organism that wants to move towards the light because they're photophilic, say. And then if they're photophilic, they'll move towards the light source. They'll seek out, they'll have like light sensors, they'll have sensory cells that detect light concentration. And when they detect a higher concentration of light, they move in that direction. So you get these gradients that increase with closeness to the source, and you get these organisms that have sensory mechanisms that allow them to move towards that source. And so a lot of times you'll see examples of chemotaxis and phototaxis where you'll have the cell or the organism will follow this path. It's not necessarily a straight path because they can't necessarily predict what the gradient will look like. But they'll explore their local area. They'll follow that gradient to the next highest concentration. So sometimes it'll look like a random walk towards the source. And so they use this to find, of course, food. They find, sources of maybe if they need to get to some point in the water column, maybe towards the top, they'll use phototaxis to do that. So that's what taxis are, and you can model taxis with things like Bratenberg vehicles. You can, in C. elegans, chemotaxis are very important for them to find food. They're always going towards a bacterial food source, so they need to be able to detect, concentrations of certain chemicals. Actually, they need to distinguish between good chemicals and poisonous chemicals, but that's not necessarily chemotaxis. That's just another part of their chemosensation. But in this case, we're talking about the minimal chemotactic cell. And so that's what they're trying to figure out what the minimal conditions are for chemotaxis. Okay, so they start with the abstract. The movement of cells and microorganisms in response to chemical gradients, which is termed chemotaxis, is fundamental to the evolution of myriad biological processes. In this work, we demonstrate that even the simplest cell-like structures are capable of chemotactic navigation. By encapsulating enzymes within lipid vesicles, and again, we're using lipid vesicles and enzymes as sort of a proxy for like this minimal cell. So we don't want to replicate we don't want to start with a eukaryotic cell. We don't even want to start with a prokaryotic or archaeal cell. We want something very minimal that kind of shows where this, how minimal can we get to get this sort of behavior. Okay, so they use lipid vesicles that incorporate a minimal number of membrane pores. So they do have pores in the membrane in this case. We revealed that a solitary vesicle can actively propel itself towards an enzyme substrate gradient. So they're simulating chemotaxis with this enzyme substrate gradient. They have these vesicles that are trying to move towards the enzyme substrate. Specifically, vesicles were loaded with either glucose oxidase or urease and embedded with corresponding transmembrane proteins that were tracked within a microfluidic device under a controlled substrate gradient. So they did this using a microfluidic device. Our findings established that the system comprising only an encapsulated enzyme and a single transmembrane pore is sufficient to initiate chemotaxis. So they are actually found that you don't need very much to initiate this chemotactic response. All you need is an encapsulated enzyme and a single transmembrane pore. So I suspect, for the chemical to get in at a certain concentration and then the enzyme to react to that. This proof of concept model underscores the minimalistic yet powerful nature of cellular navigation mechanisms, providing a previously unknown perspective on the origins and evolution of chemotactic behavior in biological systems. So they talk about the cell and how they're the minimal units of life, and how in the cells that we can see in nature today are these elaborate machines that operate under non-equilibrium conditions, but we can build these very basic cells or these minimal cells that allow us to isolate one mechanism and see how basic we can get. So we can see kind of like this ancestral form of thing that we see today, which is a wide variety of chemotaxis. Okay, so again, there are a lot of applications of synthetic biology and other areas like that. And so, in this case, they build this minimal cell where several cellular functions have been artificially recreated, including, or wait a minute, this is synthetic biology. Well, they've removed actually translation and transcription, self-reproduction, communication, and metabolism in this case. And so they need to have some mechanism for cellular transport. And among all the different motions, the autonomous propulsion of cells in response to chemical gradients or chemotaxis is the most common strategy adopted to chase nutrients, escape toxins, or control cell signaling. Okay, so this is just kind of going through how chemotaxis is sort of central to the survival of cells. Okay, so this is figure 1. This is the schematic representation of a minimal chemotactic cell. This is where we have these lipid vesicles, and they form a compartment used to encapsulate enzymes. So we have this lipid vesicle here with the alpha hemolysin pore, a single pore, and we have the enzymes inside. So you can see this, the way they've designed this. The toxic protein HLY creates pores within the lipid membrane, generating an asymmetry. So you have this single pore, which introduces this asymmetry towards the light source. So it's kind of like, it's not cheating, but it's like kind of imagining, what might help this process along. Because if we had pores all across this membrane, we might not be able to get very easily get this chemotactic behavior. So we have this, we create this pore with a toxic protein. We have this enzyme inside the lipid vesicle. So then we put this into an environment in this microfluidic device where we have water flow, and the water flow is moving it back and forth. So it's actually experiencing, it's not experiencing, the sort of totally biased system. You're generating the sort of stochasticity of the flow, and it's moving the pore around with respect to orientation. You're getting the substrate, and the substrate is coming into the cell. You get it's reacting with the enzyme here, and the products are coming out into the environment. And so eventually you want to observe where this minimal cell is moving towards the substrate source, which you do see. They have this trajectory here, and the trajectory, as you might imagine from all of this happening, is this sort of random walk, but it's randomly walking towards the source. So you're getting this kind of movement back and forth. I mean, you're not exploring all the way around the other side, but it moves towards the source pretty effectively. Okay, that's figure 1. And figure 2 shows the vesicle poration with HLY, so they're showing how they're putting holes in the, or pores in the vesicles, and they're showing that process. Figure 4 shows the drift and chemotactic velocities of vesicles and chemical gradients. So you have in A, B, and C, you have liposomes encapsulating urease. So the first thing that they used is urease. They have this velocity on Y versus channel height, and they get this. This is the measurement of HLY. And so they show this for, let's see, A, B, and C. A to C are vesicles encapsulating urease under a urea gradient of 250 micromolar per millimeter. And then D to F, which are these bottom graphs, are liposomes encapsulating GoX, which is where you have where vesicles encapsulate glucose oxidase under a glucose gradient of 50 micromolar per millimeter. Panels show A through D total drift velocity in the Y direction as a function of channel height, which is here, and then B and E total drift velocity in the X direction, and then C to F. C to F is chemotactic velocity in X, calculated by subtracting the X direction drift measured for control vesicles. So basically, this Y velocity is it moving along, or actually Y velocity is in A, B, D, and E, and then in C and F you have chemotactic velocity, and this is all kind of speaking to how it's moving in this microfluidic channel. And so it's moving back and forth, and you can see the fluctuations here. And so let's go to the discussion. So in this study, we demonstrated that only a few well-engineered elements are necessary to achieve positive chemotactic propulsion for a cell-like vesicle at the nanoscale. Specifically, we investigated whether artificial cells could use chemical gradients as an energy source to activate transport mechanisms and, under suitable conditions, be transformed into motion. So basically, they are trying to get this thing to move towards the source of the chemical, knowing kind of, what it can measure the concentration, and it can understand, or it can't understand, but it's basically measuring the concentration, and when we get a higher concentration, it moves in that direction. We don't want it to just kind of like wander around aimlessly, and if we'd had that, then we wouldn't have chemotaxis. We exploited the fact that in our body, gradients of several metabolites are very well established, so the presence or different concentration regions might affect intra and intercellular transport. We use vesicles to reproduce cell-like compartments, and we can find either glucose oxidase or urease within lipid membranes. We form few pores in the vesicle membrane to generate an asymmetry in the bilipid layer. In this way, we met the theoretical minimal conditions to achieve propulsion at very small scales. So they're not only trying to model sort of this chemotactic behavior and the conditions for chemotaxis, the mechanism, but they're also trying to model this sort of propulsion. How do you achieve propulsion? And so that's how they, in this model system, they're able to do that as well. When placed in chemical gradients of the respective substrate, enzyme encapsulating vesicles show a chemotactic component towards the substrate source, confirming the ability to autonomously propel. Thus, the confinement of chemical reactions in nanometric volumes was able to generate energy sufficient to be transformed into autonomous motion. So this is a proof of concept, and it demonstrates that a cell-like vesicle equipped with confined active molecules and a transmembrane protein can effectively overcome Brownian motion and be chemotactic. So we don't want Brownian motion where it's moving around the source kind of in that way that we saw. It's because it's kind of trying to find the next highest concentration of the chemical. It's going to move around sort of in a Brownian motion towards the source, but we don't want Brownian motion where it's just kind of orbiting the source and just kind of, totally lost in space. Okay, so these results are promising from a biochemical point of view because the elements we studied are ubiquitously present in the structure of a large majority of cells. For this reason, we are convinced that understanding in more detail the behavior of our system will significantly improve the comprehension of complex biological phenomena, such as vesicle transport, disclosing mechanisms that are not fully understood. So that is that paper. So let's go to the third one. And I'm not going to get deeply into this, but I did want to bring this up in the same context as these other papers. So this paper is from the archive. This is from last year, so it might be published somewhere by now, but this is thermodynamic space of chemical reaction networks. So this describes a chemical reaction network and then the accompanying thermodynamic space. The abstract reads, living systems are usually maintained out of equilibrium and exhibit complex dynamical behaviors. So if we're talking about vesicles, these are systems that are equilibrium systems. We can create them in different ways that are very simple, and we can understand their outputs, and it's pretty good. But so in more complex cells, like the cells that we observe in living systems, bacterial cells, archaeal cells, especially eukaryotic cells, these systems are maintained out of equilibrium. That means that they often exhibit out of equilibrium behavior, and so we can't use equilibrium models to really understand what they're doing. We need to understand, they exhibit these kind of complex dynamical behaviors over time that we need a different model for. The external energy supply often comes from chemical fluxes that can keep some species concentrations constant. So this is in chemistry where we have different types of chemical species, and they have to be sort of, they're either in equilibrium or more often than not, they're going to be out of equilibrium. But nevertheless, you need to have constant concentrations of things to have reactions. Furthermore, the properties of the underlying chemical reaction networks, or CRNs, so I talked about GRNs, which are genetic regulatory networks. These are chemical reaction networks, or CRNs, and they operate in a similar way. Basically, they're connected by, what chemical reactions are happening, which chemical reactions other chemical reactions enable, and so forth. I'm not as familiar with those as I am with GRNs, but there's a caveat here. So the properties of the underlying chemical reaction networks are also instrumental in establishing robust biological function. Hence, capturing the emergent complexity of living systems and the role of their non-equilibrium nature is fundamental to uncover constraints and properties of the CRNs underpinning their functions. In particular, while kinetics play a key role in shaping detailed dynamical phenomena, the range of operations of any CRN must be fundamentally constrained by thermodynamics, as they necessarily operate within a given energy budget. So we need to have this sort of thermodynamic constraints in our model, and so that's what they're talking about here. Here we derive a universal thermodynamic upper and lower bound for the accessible space of species concentrations in a generic chemical reaction network. So what they're doing is they're basically creating this kind of model where you have basically this possibility space, and they're trying to figure out what spaces are accessible and inaccessible given the thermodynamic limitations of the system that we're talking about. So all we need is energy. The more energy we have, the more of that space we can access. The less energy we have available, the less of that space we can access. The resulting region determines the thermodynamic space of the chemical reaction network, a concept we introduce in this work. And so this is kind of like, if you think about like a combinatorial system where you have a lot of different things interacting and you have so many different combinations, and you can use a mathematical model to describe that space. that space isn't limitless. Sometimes it's constrained by different things. It's constrained by plausible combinations of things. It's constrained sometimes by thermodynamics. Sometimes it's constrained by the number of steps you can take. And so that gives you this plausible space of things in that larger space. And so that's kind of how they're thinking about this. Moreover, we obtain similar bounds also for the affinities, shedding light on how global thermodynamic properties can limit local non-equilibrium quantities. We illustrate our results in two paradigmatic examples. The first is the Schlegel model for bistability, and then the second is a minimal self-assembly process. So bistability is where you have a system where there are two stable states, and you transition between those two stable states. So if you have if you're in one stable state, you might operate in that stable state for a while until you hit a critical point, and then you transition into the other state. And you go back and forth between those two states. You stay in that stable state for a while, and then there's some point at which you transition to the other state. There are a lot of systems that are bistable. Sometimes they're on-off switches. Sometimes they transition between two states of behavior and the like. And then, of course, minimal self-assembly is where we want to find the minimal conditions for self-assembly. What are the thermodynamic constraints of that process? So both of these cases demonstrate how the onset of complex behaviors is intimately tangled with the presence of non-equilibrium driving. In summary, our work unveils the exact form of the accessible space in which a chemical reaction network must work as a function of its energy budget, shedding light on the non-equilibrium origin of a variety of phenomena, from amplification to pattern formation. Ultimately, by providing a general tool for analyzing chemical reaction networks, the presented framework constitutes a stepping stone to deepen our ability to predict complex out of equilibrium behaviors and design artificial chemical reaction systems. So this is good stuff. A lot of complexity theory here, a lot of biophysics. And so they just make the point that living systems operate far from equilibrium, consuming energy to maintain organized states against the tendency towards increasing entropy. So this is an interesting point that entropy usually acts to degrade a lot of processes. So when you're talking about a minimal chemical system, you're talking about a system that is probably constrained quite heavily by entropy. Whereas living systems, they don't defy entropy, but they build a lot of complexity in spite of entropy. And so to do that, they have to operate far from equilibrium. They have to consume energy in different ways, and they have to maintain these organized states. And a lot of this is very interesting because we often think of an entropy and thermodynamic constraints in an equilibrium sense or in a closed system sense. So it's cells in living systems are open systems, meaning that you don't close off or you don't limit the amount of input energy. They're getting input energy from a lot of places. And so there's a different set of rules, for open systems than there are for closed systems. Chemical reaction networks, or CRNs, capture many of these biochemical processes, including gene regulation, metabolism, and cellular information processing. To analyze these complex systems, chemical reaction network theory has been developed, providing a powerful framework for characterizing CRNs and revealing how complex behaviors emerge from underlying molecular interactions. And so the complexity manifests in various forms, including what they call non-equilibrium stationary states, multi-stationarity, chaos, and oscillations. So these are the different ways in which you can get different complexity-related behaviors. None of these are, reliant on the sort of equilibrium model. The wide range of possibilities is instrumental for establishing robust biological functions, together with the necessity of continuously harvesting, dissipating energy into the environment. So they need to get energy from a lot of places. They need to move energy around different processes, and there's a wide variety of processes, robust biological functions that need to be established in a living cell. And so these non-equilibrium conditions play a role in this. So they can amplify the abundance of a desired biochemical state, trigger the onset of complex dynamics, and regulate the emergence of large-scale structures. And so they kind of get into this landscape or possibility model where they model all the possible states, and then they kind of constrain it given the amount of energy coming in. They do the math here. They provide some calculation of the different chemical species in the chemical reaction network. And so they work out the thermodynamics and geometry of these networks in this section. And then they talk about the universal thermodynamic bounds for chemical reaction networks. So we have these thermodynamic accessible regions. We have this thermodynamic space that emerges. So these are the accessible regions for species concentrations. And then they connect this to previous results. So this math just doesn't exist as this model. It actually connects to what people are doing in the field and what's going on in the real world. So this shows the Schlegel model, which they talk about in the abstract. This is the thermodynamic space of the Schlegel model. So this is the bistability model that we talked about, where you have stable points and then unstable points. So your stable states are A and B. These are two chemostatic external species. And then you have this system. I guess this is a hypergraph model. So you have these two chemical species. You have this concentration X, and then when you get to a certain concentration, you go from A to B. As you hit this unstable region of concentration, you go into B. And then the same thing happens at B, you go into A. And so you can see, actually, I think this is a one-way reaction. So this is like A is the stable state here, and the concentration is here. This is the sort of the differential of that function. And then you hit this unstable point, and then you transition quickly to the other state B. And then that exists in a stable state. For B, you have this, they call it EFM of the Schlegel model, in which the internal species X is created from the environment, which is in green here, and then degraded into it respectively through reactions 1 and 2. So we have these chemical reactions. This is chemical reaction 1, this is chemical reaction 2. In A, it enables this move to an unstable state, then also enables a move back to a stable state. In B, we have this, we have the environment having an effect on this internal chemical species, and then these two reactions, either a direct path or an indirect path, that transform this, and we get this nonlinear function here to show for it. Okay, so then they kind of go through the Schlegel model and the mathematics of it. This is the reaction hypergraph with 3 internal species, 2 external species, F and W, which are in the green, and then a chemical probe, which is in dashed red, which is on the outside here. And then for B, the elementary flux model, or EFM, including the chemical probe, with vector representation E hat. So we have this represented in this way. A sequence of reactions from the EFM, converting species from left to right of the probe. Thermodynamics for D, this is the thermodynamic space here, which is this plane on this 3-dimensional space, and then the conservation laws, which are this triangular plane on the space. And there's this intersection of the yellow and the green, which is the thermodynamic space given the conservation laws. That's where your steady state concentrations lie. So the idea is you run these networks and you show kind of what the thermodynamic space is, you use the conservation laws to constrain that thermodynamic space, and then you get this surface or this manifold of the possible steady state concentrations for these species, F and W. Then they talk about self-assembly and the mathematics behind that. So the discussion basically concludes that we have derived fundamental thermodynamic constraints on generic chemical reaction networks at stationarity, introducing the novel concept of thermodynamic space to describe the feasible range of chemical species concentrations compatible with the system's energy budget. Our approach is based on chemical reaction network hypergraph geometry and utilizes the novel chemical probe technique to study chemical species that are directly linked by an existing reaction. So it significantly extends and generalizes previous findings on linear and catalytic networks. So there's this area of pseudo-unimolecular chemical reaction networks to arbitrary network topologies, and that's one example of these kind of linear and catalytic networks. This generation is crucial for applications in complex multimolecular chemical reaction networks, such as multi-stationary biochemical networks and self-assembly processes, which are widespread in living systems, but their study has fallen short of a comprehensive thermodynamic analysis. And that's what they've introduced here. So that's all I have on those papers. I wanted to present those as a group because I think it kind of gets at some of these things we've been talking about with early life, but also with these kind of models of biologic or biophysical space. How do we represent all the possible outcomes of a complex system? And then how do we evaluate that? Comments or questions?
How does that fit in with organelles in a cell? Because it's some sort of a similar process from what I'm seeing. Like the organelles develop kind of on their own within a cell. Like they're some sort of a separation occurs inside a cell due to the chemical nature of the chemicals inside.
Yeah.
They separate into organelles.
Yeah, I'm not sure. I don't know if there are any papers. I might try to find a paper for next week on that.
Yeah, I have some.
Oh, okay, you do? All right, well,
I'll
send
try
them
to
along,
find
yeah.
them for you.
All right. Yeah, because I'm not sure what the answer to that is. Yeah, I'm not familiar with the development of organelles, but it sounds like you do, so we'll check it out.
Well, I was interested in mitochondria.
Yeah, mitochondria are interesting in that respect.
Yeah, I don't know. I haven't really read them. I just collected the papers.
Yeah. All right, thank you. Any other comments or questions?
Oh, did we lose him?
I'm here.
Okay. Are those synthetic cells? Are they? And I know cytoplasm, but like, are they able to produce more of their cytoplasms, or as they divide, are they continually getting smaller and smaller? Phobic exterior and hydrophilic interior that they mentioned, but
yeah,
they divide. Are they getting smaller and smaller, or are they actually like reproducing, if that makes sense?
Yeah, like in a lot of cells, they'll get smaller and smaller as they divide, or they divide into half the size, and then they might grow like, as newly reproduced cells. In vesicles, I guess it depends on how they set up the system. So a lot of vesicles have a deterministic size, and then they sometimes they're doing things like shedding amphiphiles, and they're kind of creating a new vesicle at a critical size. Sometimes it's a fixed size. I'm not really familiar with the ways you can do this. I mean, there are probably a lot of techniques that you can use to sort of, replicate what goes on in biology, which is the point. But yeah, I don't know.
Okay, thank you. Also, and I'll put the link in the chat. If you have Springer access, there is a book on CRNs that you can get for free.
Oh, great. Yeah, thank you.
So
Yeah.
I imagine most of us here have some sort of institutional access. So.
Before we go, are there any updates that people want to give? I don't know if Hussain had an update or.
Sure. Yeah, no problem. Okay. So, a bunch of different projects. The C3 projects. I think I might have mentioned this before, but I'll just go over it again because it's. I don't know who was here. I don't know if. Yeah. So, we've made really tremendous progress over the past couple of weeks on the RBRCT. So that's the ray-by-ray computed tomography project to reproduce images using CT technology and show that the combination of the MART algorithm, which Dick first proposed back in the 70s, the multiplicative outbreak and reconstruction technique, the MART algorithm could reconstruct images with very little information. And all of this is based off of CT approaches, with our new setup and apparatus showing that we can that we can steer X-rays electronically on a very fine-tuned, precise level. The gist for this update is that I finally got the MART algorithm working with the ASTRA toolbox. The ASTRA toolbox is the set of photon scattering, light technology, light optics sort of programming software scripts that we're supposed to be using. Okay. Previously, we had tried implementing things from basic Python and MATLAB and stuff from the ground up, which may have worked, but to actually show that this is feasible and presentable, we need to be able to use what Mohammed Majid Al Rafai, so he's the computer scientist over in UK, what he himself had recommended. Yep. So that's the biggest update on that side of things. In terms of the work between me and Dick and Savina with Savina's group, so that's on the peptide. That's on the peptide project. We are studying vesicle formation with peptides across the lipid membranes to see how they may have come about, and that was kind of in reference to the paper that Bradley had just mentioned, where the growth and reorganization of fully abiotic artificial and synthetic cells. That paper, which supposedly some people had called the most important paper of 2025, which once again, so that's a pretty bold one, and that's a pretty bold one. But we're writing a bunch of different papers and doing simulations based off empirical data, notwithstanding like the book The Laws of the Game, which I just got through the mail recently, so I'm reading through that one. But at its root, we are talking about imagining lipid membranes also as sort of a chessboard, where you have L membranes and D membranes. L and D membrane peptides from mixed LD peptides, and the basic question is, how do peptide membranes form from that? Okay, can you show based off entropy, based off thermodynamics, based off the simulations from empirical results between computational data approaches, that the actual membranes of cells are able to reasonably and visibly form based off the empirical information and from the origin of life in and of itself. So really exciting stuff, and this just affects basically more or less every different area you can come across. And yeah. Excuse me. In other news, I recently learned, speaking of evolutionary approaches to the origins of life and things, I recently learned that whales descended from land mammals, and that was really surprising, because I was for whatever reason under the impression that life went from one direction, from sea to the land, and I totally forgot about that. That there are aquatic creatures, fully aquatic creatures, which also descended from land animals themselves, which sort of went back into the water, and that's pretty surprising. So whales are more closely related to like horses and wolves, and
to the best of my understanding, any mammal that is in the sea went from land to sea. Although I could be wrong about that?
No. I thought it was typically unidirectional in one direction. Like anything that is walking on land and spends more or less all of its life on land originally started out 100% completely aquatic underwater. You know, but then I realized things like whales. Nope, they went the other direction. Yeah. It's based off the vestibular fins. The bony structure in their vestibular fins matches much more closely the structure of horses. And not necessarily saying modern day horses, for whatever reason, started jumping back in the water for no reason, but more so the common ancestor between horses and whales shows them much more closely in that, much more closely in that direction too. So yeah. That's just something interesting I came across too. So.
Yeah, that's good. Okay, I think that's it for today. Thanks for the update, Hussain, and I'll post the papers.
To put things in perspective, the ASTRA toolbox, so that's what we use for the CT computer tomography approaches. It has taken me 4 months to get it running.
Wow.
4 months to get it running, to get the algorithm coded properly. Not just based off technical codes and errors and issues and things, but it may seem just like very, very straightforward, but the toolboxes, they themselves have their own quirks and idiosyncrasies you have to plow through. And even Mohammed himself, if he had been using Astra for a long time, he still spent quite a bit of time getting it to work. So
yeah,
one thing for me on a daily basis when it comes to just like technical things and complicated stuff, it can be difficult to really sit down and get focused on the precise errors about what's going wrong. Many times it's just so useful for, especially for us academics, to kind of just like black box sit and say whatever happens happens.
Yeah.
When because if we couldn't show that the Astra toolbox could be used to show the MART algorithm could reconstruct images, then we had no basis. There's no way any journal would publish that. So. Yeah.
Well, that's good. Yeah. All right. Thank you. All right. That's it for today. I'll put the papers up on the YouTube description, or probably in the Slack as well. And thank you. See you next week. Bye.
Okay, bye.
Bye.